Hello, how are you?
I'm good. How are you?
Very good.
Yeah, it's nice for you to join me on the podcast. I'm really excited.
Oh, no, thank you very much for the invitation. This is great.
Yeah. So I'm Ivanka. I'm a high school student from Minnesota, and I'm really passionate about climate and sustainability. And I'm really interested in methane emissions specifically because I did my project at the University of Minnesota under a scientist, Dr. Joe Hoisley from the USDA. So I did my project on reducing methane emissions from livestock through feed additives. And then I did a similar project at Cornell. And so I started this initiative, MethaneMatters, to help educate people on methane emissions, like their sources and impacts and mitigation strategies. And yeah, I'm doing these podcasts to just educate people. And also I wrote this book to that goes a lot more into detail than my videos on this channel. Yeah, I'm really excited. This is like I've had a lot of podcasts so far, so I'm really excited to have you on the podcast.
That's excellent. Tell me, and where are you going for college?
Oh, I'm applying to like UC Berkeley and Michigan. I'm also
Okay.
applying to Wisconsin Madison because yeah, it's nice. It's only a few hours away, so that's good.
Yes, yes, it is. That's good. That's good.
Yeah, yeah. So how about you? What do you do?
Well, my name is Francisco Penagaricano. I'm an associate professor in quantitative genomics here at the University of Wisconsin-Madison. I'm in the Department of Animal and Dairy Sciences.
Okay. So, and then my first question is, what's like your educational background and how did you get started in your career?
Okay, yeah, that one is a good one. So I'm originally from Uruguay, this small country in South America. Um, so I received a BS in biology and one in biochemistry back in Uruguay. And also I did a master's in animal breeding and genetics. And I came to US in 2010, actually to the University of Wisconsin-Madison, where I got my PhD in animal science. And when I was doing my PhD, I also did a master's in statistics because I had the free time, you know. No, I'm kidding. Um, so I finished in when was December 20, 2014. I finished my PhD, and then I was 5 years at the University of Florida. I was faculty in the Department of Animal Sciences,
Okay.
and then I moved back to Wisconsin in August 2020. Um, so we are what? It's almost 5 years.
Yeah, more than 5 years now.
Yeah, you're right.
Okay. So, how did you get interested in the field you're in currently?
So I'm part of multidisciplinary, multi-institutional group of dairy cattle researchers uh, from four land-grant universities. It's University of Wisconsin-Madison, University of Florida, Iowa State, and Michigan State, and also the USDA and the Council on Dairy Cattle Breeding. So
Okay.
we are geneticists, we are nutritionists, we are microbiologists, and we have been working together for, I would say, almost 15 years.
Okay.
We started working on dairy cow feed efficiency, and a few years ago we started to work on methane emissions, as you know, and I think it was a natural next step after to work with the feed efficiency to actually work on methane emissions, and that's why probably 3 years ago we started to work on methane emissions.
Okay. So can you tell me about some of the projects you're currently doing?
Yes, absolutely. So first of all, we are phenotyping. That means in our field basically to uh, uh, measure, quantify a given trait. So we are measuring methane emissions on lactating dairy cows. We basically do it in mid lactation
Okay.
when the cows are between 50 days in milk up to 200 days in milk. And we are doing that on our research farms. And we are also doing that on commercial farms.
Okay.
Um, we are also measuring methane emissions on young bulls, and we are doing that on bull stats. So we have an ongoing collaboration with Genus ABS and with Urus Pig Genetics. That's at the level of uh, quantifying emissions, right? And for that we use the green feed system, and
Okay.
with that we have um, uh, one of the beauties of the system is we can expose cows 24/7 for 7-8 weeks to the system, so the cow go and visit the system whenever she wants, and then we measure grams per day directly with the system, right?
Okay. Yeah.
And then
With the green feed system
yes,
I was at Cornell.
yes, okay. So then we have three main goals in the project. Um, and those goals are based on the following problem that we have right now. If we visit a farm, um, if we go together this afternoon and visit a farm, right, a dairy farm, and we enter in a pen, let's say we enter in a pen with 80 cows, right?
Okay.
And then you ask me which cow is producing most methane in this pen,
Mhm.
or who is the cow that is producing the least amount of methane in this pen. Um, Basically, today we do not have an accurate tool to predict methane emissions.
Okay.
We can use, if you tell me the lactation and milk production and probably body weight, we can kind of try to rank the cows, but we don't have a way to do it. So we are trying to develop a predictive tool so farmers can have an accurate estimate of who are the cows that are producing more methane, who are the cows that are producing less methane in a farm. And we are doing that through the milk spectra data.
Mhm.
Um, So farmers in this country, they typically test milk once per month, right? They sample milk, they send to a milk lab, and with that, they have an idea of fat, protein, lactose, and even somatic cell count in the milk. And we are trying to use the same methodology to actually predict methane emissions. So farmers, when they test their milk, they will have also an idea of methane production per cow.
Okay.
This is one. The second one is if we are in the same farm and the farmer tell us that they want to breed cows for reduced methane emissions, we do not have the tools in this country. So
Mhm.
what we are doing is we are developing genetic tools so farmers can breed cows for reduced methane emissions. We did that as a team for feed efficiency, and now we are doing the same basically, but for methane emissions.
Okay. So which genetic tools are you using?
So basically, we call genetic tools as national genetic evaluation, right?
Okay.
So you can actually estimate the breeding value of an animal that basically tell you how good will be the offspring of that animal, and that's what I'm calling now a genetic tool to be able to select. And you know, farmers in this country, they don't select for a few traits. Uh, Farmers use economic selection indices to actually select animals, and our plan is to develop the evaluation and facilitate the implementation of the evaluation in, for example, the Net Merit Index. That is probably the flagship of the indices in the industry. So we are working on that. And the third one is the third goal, right? The first goal is develop kind of I call a management tool to be able to predict what are the cows that are producing more or less methane in a pen. The second one is to develop a national genetic evaluation so farmers can breed to reduce methane emissions. And the third one, you know, at the end of the day, methane is produced by the rumen microbiome. So we need to have a better understanding of how the composition and function of that microbiome affect methane production. So for that, we are investigating the rumen microbiome, right? But actually, to have access to the rumen microbiome is complicated, right? We use a procedure that is called tubing, where you actually sample the rumen content of a cow. But we do that only on our research farms and on few animals. So we are interrogating two other microbiomes that we believe they can be correlated with the rumen microbiome.
Okay.
And we can use those microbiomes because they are easy to access, and then we can think in more high throughput methods, right? And we are using the two extremes. We are sampling the oral microbiome as a proxy of the rumen microbiome, and we are sampling the fecal microbiome also as a proxy of rumen microbiome. And we have some very cool result with the fecal microbiome right now.
Okay. So do you ever use like rumen modeling for the microbiome?
We do. We have some very cool papers of rumen microbiome and feed efficiency, but probably it's exactly the same with methane. If you think about a given trait, let's say methane emissions, right? You have kind of a triangle between the host genome, if you will, the rumen microbiome, and the methane production, right? And then you will have the host affect methane production, the host affect rumen microbiome, and the rumen microbiome also affect methane production. So that system, we typically model that system using structural equation models. That is it's a system that allows you to have predictors that are sometimes response variables. So it sounds complicated, but it is not. So we have been modeling rumen microbiome using those systems. Basically, those are based on networks.
Okay. So do you do any simulation or computation models?
We do all the time. That's what we do. Yeah. So so far we have been estimating the heritability and repeatability and genetic correlations of the methane emission traits. That's what a geneticist will do in the first step. And I can tell you, methane production is heritable. Is probably more heritable than meal production. That is quite important. We have been working also on residual methane traits, that is methane production adjusted for example, meal production or body weight or intake, right? Um, it's this idea of if I have two cows that produce the same amount of milk and they have the same body weight, the cow that is producing less methane is more environmentally friendly, right? And also probably more efficient. It's this idea to try to have two cows at the same level. That's why you adjust for production and body weight.
Yeah.
So those residual traits are also heritable, around 20%. What's our great
Okay.
news?
What do you mean by residual traits?
Well, residual comes from a model.
Okay.
So we first adjust a model where we have production as a response variable, and then we have meal production and body weight as predictors, and then we get the residuals from that model, and we use that as a new trait. That's why it's called residual traits. Yes.
Okay.
And then probably one of the most important findings is that methane production and feed efficiency are positively correlated. That means cows that tend to be more feed efficient tend to produce less methane. Those are great news, right? Because we as an industry, we have been selecting for feed efficiency. We do have an evaluation that was implemented in December 2020. So we have been selecting indirectly for methane emissions, and now we will have a trait to select directly also for methane emissions.
Okay.
So this is in terms of genetic parameters, right? This effort that we have to develop genetic tools. The second one is the use of a spectra to predict methane. I told you, right? As a management tool.
Yeah.
And we have very promising results with very, I would say, very high correlations. Even when you train a model in one farm and you try to predict a completely new farm. So I think we will be able to have a predictive tool based on spectra that farmers can use to have an idea of who are the cows that are producing more methane, for example.
Okay.
And then, well, this is all about modeling, right? We do models all the time. And then, for example, for the genetic parameters, those are classical models in animal genetics. Now, for spectra, we are using some machine learning methods. You can use models as kernel models, for example. So you use multiple sources of information. And then for the microbiome, we have been exploring that triangle that I mentioned before, from a classical mixed model approach to using some structural equation models, even to use some networks. It's fascinating, right? This idea that you have a triangular system and how you model all those effects together.
Yeah. So going back to the point where you talked about feed efficiency and the correlation between methane emissions,
Yes.
does that also correlate in some way to the milk production of the cow?
So, okay, let me go back. The idea of feed efficiency is the following. If I have two cows that produce the same amount of milk and they have the same body weight, the cow that is eating less is more feed efficient, right? Because she's producing the same, eating less. That cow has a negative residual feed intake, if you want. Residual feed intake, that is also a residual trait, is the way that we use to measure feed efficiency.
Okay.
So what we found that is super cool is the cows that have negative residual feed intake, that means that are more feed efficient, tend to produce less methane.
Okay.
So it's a win-win. If you think about two sustainable traits, to be feed efficient and to produce less methane, those traits are correlated in the same direction. So if we push both, we will move faster, right?
Yeah. Okay. So they eat less and they produce less methane.
Yes.
for the same output. That makes sense.
Yes. Now, if we just go to the farm, remember we were visiting a farm this afternoon? If we go to that farm and we select the cows that are producing more milk,
Mm-hmm.
probably some of those cows are the cows that are producing more methane in the farm. Why? Because cows that tend to produce more milk, they also tend to eat more. Probably they will produce more methane, right?
Okay.
That means methane production and milk production, they are positively correlated.
Mm-hmm.
When we use this idea of residual trait, we adjust for production in order to have two cows in the same level, right? They
Yeah.
produce the same, they have the same body weight. Now I can compare their methane production levels.
Okay. Okay. That makes sense. And then what is your like lab composition and who all do you work with?
Oh, they do all the work. I'm just the cheerleader, actually. We have a very large group. We have 2 or 3 or 4 postdocs. We have 3 or 4 research specialists. We have 4 or 5 graduate students, and we tend to have always some visiting scientists, mostly students from other countries that come and they spend maybe 3 months, 6 months, a year with us, learning and working here with us. So we are a very large group, and we are quite diverse and multicultural. So I'm from Uruguay. Um, we have people from Uruguay, people from Argentina, people from Brazil. We have from US. We have from Iran. We have from Turkey. We have former students from China. All over the world.
So are you doing any like international projects in these countries?
That's a very good question. I have a very active collaboration with Uruguay.
Okay.
And I collaborate in a project there right now in methane emissions. The system in Uruguay it's a quite different because it's kind of a grazing based system, right? So where the cows actually are outside grazing. So the dairy industry is a bit different. So I'm collaborating in methane emissions in Uruguay right now. Similar project that we have here, but with other challenges, if you will.
Okay. So what are those challenges?
Well, for example, is how to measure methane emissions in a grazing system, right? So basically, in a grazing system, if you have the green feed right outside, that's one approach. Probably you won't get too many visits.
Okay.
So another system or another way is, okay, if I have a robot, I can use a sniffer, right? The sniffers, they have other challenges, right? For example, the sniffers, they don't measure grams per day. They measure concentration. So then if you need to have grams per day, I mean, you need to massage the data. So, different systems, different challenges, but at the end, the same goal, right? To develop tools so farmers can, as a whole, we can mitigate methane emissions.
Okay. And then I'm curious if you're doing any projects in India or anything around there, because they also have a different system that like
They do. No, I'm not. No,
Okay.
I'm not. but more than happy to have one. So if someone is watching this and they have an idea and funds, more than happy to help.
That's good to know. Okay, yeah, what other like technologies are you using? I know you talked a lot about this, but like kind of like a summary of all that.
Well, all the cows that we are phenotyping, right? We use this term phenotyping that basically means measuring, right?
Hmm.
All the cows that we are phenotyping, they are genotype.
Okay.
So we do have genomic information for those cows, roughly 70,000 markers across the genome. And that's very standard in what we do. We have genomic information.
Okay.
Then, in terms of how we measure methane, I told you we use the green feed system. The idea of develop a predictive tools is based on spectroscopy, right? That is actually the method that we use to actually quantify or evaluate meal composition. And then in order to better understand composition and activity of the microbiome, what we are doing is next generation sequencing. So we are using two approaches. We are using the famous 16S that to evaluate composition, and we are also doing some long read sequencing of the rumen microbiome. So we are combining different sequencing strategies there. So we do, let's say, full bag of technologies, from classical genotyping of cows. In our research farms, we also measure intake. So we use some cool blue gates, or we have Callen gates, depending on the farm, how we measure. So think about it. For feed efficiency, you need to know exactly how much a cow eats every day, right? So
Yeah.
in order to do that, you need to have kind of equipment to measure that. You need to know exactly if this cow eats 25 kilos of dry matter intake, of she eats 23, of she eats 29, 30, right? You need to quantify that. And then I told you, you need to have an idea of milk production that we have with the equipment in the parlor, but we also need to have an idea of body weight, right? In order to adjust for that. So we also need to weigh cows regularly.
Okay. So at UW-Madison, how many cows do you have?
So in the university, we have 3 research farms.
Okay.
So we have one that is here crossing the street, here right in the middle of the campus, in the middle of the city. That's the dairy cattle center. And we have there 80 cows.
Okay.
My group, we don't do research there. That facility here is mostly for teaching, but mostly for very intensive research project, right? Typically done by my friends, the physiologists, right? That you need to infuse cows and take samples. So there we have 80 cows. Then 45 minutes from here, we have the Arlington Research Station.
Okay.
We have a research farm there where we milk roughly 600 cows. And in that farm, we are evaluating 2 groups of cows every time. So we have one pen where we evaluate 64 cows, and we do methane emissions and intake daily. And we
Okay.
do that for 7-8 weeks. And we have another pen where we typically do it kind of resemble of a commercial farm. We do only methane emissions there with 32 cows. And then 2 hours, 30 minutes north here, we have Marshfield Research Station. This is our third research farm. There we have our heifers and some of our first lactation cows.
Okay.
We typically milk 150, 200 cows there, and we run there one pen all the time with 32 cows, and we do also intake and methane emissions. And we do that group of cows, we run it for 7-8 weeks.
Okay. So do you ever collaborate with any other universities nearby, like the U of M or anything?
Yes. Okay. So part of this project we have Iowa State University. Iowa has a research fund, and Iowa is collecting data for this project, intake and methane and other traits. We collaborate with the University of Florida. The University of Florida has also a research farm, and they do the same. And we collaborate with the Michigan State University. They also have a research farm. They have actually a brand new research farm, and they do the same. And we collaborate with the USDA. The USDA, the Dairy Forage Research Center, is just crossing the street here in front of my office. They do have a research farm here in Wisconsin, and they are also collecting data, intake and methane emissions. So yes, we collaborate with other groups too.
Okay. Yeah, I'm doing my research project right now at the U of M with Dr. Joe Hoishley from the USDA.
Mm-hmm.
Yeah. Do you know her by any chance?
No. Where is he?
She's at the University of Minnesota.
Okay.
Yeah, with the USDA.
But it's USDA.
Yeah.
Yeah.
Yeah. Do you collaborate with any like dairy farms?
And then yes, that's a great question. Then we are right now phenotyping. That means measuring methane emissions in 3 commercial farms here in the state of Wisconsin, and we are trying to have more. So in one, we have 2 green feeds in one farm, 2 green feeds in another farm, and 1 green feed in another farm. So we are right now working in 3 commercial farms. And I think I mentioned at the beginning, we are also measuring young bulls. And for that, we are collaborating with 2 companies, 2 big companies, Genus ABS and with Urus Peak Genetics. Both companies, they have their bull facilities here in Wisconsin. So we have green feeds in both stations, and we are measuring emissions in bulls. We do it in bulls between 5 and 6 months of age.
Okay.
And the question that we have is very simple, but it's extremely important. We want to know what's the correlation between methane emissions of a young bull and methane emissions of a lactating cow.
Okay.
I go again. What's the correlation between methane emissions of a young bull, and then you can imagine what is the correlation between his emissions as young bull and the emissions of his daughters when they are producing milk. That's basically the question that we want to answer. If the correlation is high, that could be a game changer. Why? Well, in this country, we have 9 million dairy cows.
Okay.
But actually, we screen maybe 2,000, 3,000 bulls per year. So if the emissions of a young bull are highly correlated with the potential emissions of his daughters, then maybe it's easier to phenotype the bulls instead of the cows.
Okay.
Because actually we have very few bulls compared with cows.
Yeah. So you'd have to test less of them.
Exactly. So that's what we are doing. And this is again is a collaboration with ABS and a collaboration with Pig Genetics.
Okay. Yeah, so would you have any advice for a high school student or someone like me who would
Well,
want?
I really love what you are doing. I'm extremely impressed of what you have been done, and I after you invite me, I Google you, and you are everywhere with the podcast and books and whatever. I think it's amazing, and um, I think what you are doing is what other students need to do to be involved, right? And it will be great if you come to UW Madison.
Yeah, I would love to. Yeah, thanks for taking the time to join me on my podcast.
No, and thank you again for the invitation. It was great.
Yeah, it was really fun.